Good evening, everyone. Madam Winnie here. Thank you so much for the subscription. Thank you so much for the support. Still, I urge everyone to subscribe, whoever haven't, so that we can find new things and we'll be able to continue learning. Today, I'm going to introduce metabolism. We've done biomolecules. If you check any of the sessions of the videos which I've uploaded, we've done carbohydrates, we've done proteins. I've not yet done fatty acids, but I thought since I'm already done with the carbohydrates, it is better to do metabolism, that is carbohydrate metabolism to be specific, so that students can also try to understand how the metabolic pathways of the biomolecules, which we've so far discussed, is also very important, and they can also try to understand how all those metabolic pathways in the carbohydrate metabolism are important to any of those organisms or the living organisms. So as much as we introduce much on the biomolecules, or in the introduction I talked about the cell as the basic unit of life, I can remember also mentioning in the introduction slides that when cell is a basic unit of life, but in the cell there are subcellular components, which in other words are known as organelles, and in the organelles the metabolic pathways or reactions take place, and that is what makes life complete. That is all we can understand about the cell, the basic unit of life. Life will be there when there is a reaction which is taking place. So the biomolecules already forming the components of the organelles, but then inside the organelles there are chemical reactions taking place. So the chemical reactions which take place in those organelles is what we are going to discuss today, and we see how important the chemical processes are to those living organisms. So I'm going to start with the simple ones, the carbohydrate metabolism, and I'm going to start by diving into glycolysis. So when does glycolysis take place? What is the definition of glycolysis or glycolysis, in other words? So if you look at it, glyco is glucose, or basically what we mean is a carbohydrate component is in there. Then lysis is breakdown. So we are breaking our glucose to pyruvate with the main objective of generating energy in form of adenosine triphosphate or ATP. But let's ask ourselves that at what point do glycolysis take place? At what point are we breaking down glucose? Glycolysis, as one of the metabolic pathways, is always taking place when there is excess glucose. Levels of glucose is high. And if we talk much about glycolysis, that is degradation. So generally when we talk about metabolism, it involves 5 processes. There is digestion, there is absorption, there is degradation and synthesis, and lastly there is excretion. So before the glucose gets broken down by the glycolysis process, then whatever happens first is we have to ask ourselves immediately you take your starch in the morning. It could be bread, it could be potatoes, any component of a carbohydrate, then digestion has to come first. So digestion will start from the mouth. The salivary amylase working at a pH of almost neutral, 6.5, 6.7. Then, after the mouth, you are breaking it down by chewing. Then it will pass the stomach. Because of the acidic pH of the stomach, carbohydrates are not going to be digested in the stomach. So the pH at 1.7 is very, very acidic. So the carbohydrates cannot be digested there. Then it will move to the small intestine. In the small intestine, it will further be broken down. If it was amylose, for example, we already saw the examples of the polysaccharides where we saw starch as one of the homopolysaccharide. If you refer to the slides of polysaccharides, we'll understand that. So it is going to be broken down to simple glucose units. So it was just a homopolysaccharide, only one monosaccharide component. Our polysaccharide dehydrolyzed only one type of monosaccharide component. So in the small intestine, the maltase, which is a hydrolase, if you look at the class of enzymes, as we discussed in class, so the maltase, which is hydrolase, will break the maltose into 2 glucose units. So after digestion, just to move a little bit faster by not going into details, whatever happens, then after it has been broken down to 2 glucose units, then absorption takes place. Absorption of glucose will take place by active transport. So glucose absorption is different from fructose absorption, because fructose, being the second carbon has a keto group, it will be absorbed slower by facilitated diffusion. But for the aldoses, they'll always be absorbed by active transport. It means even by facilitated diffusion, they can be absorbed by that. But fructose cannot be by active transport because of the second carbon having the keto group. So its absorption is a bit slower. So our glucose is going to be absorbed. Then after absorption is when glucose oxidation or glycolysis takes place. So we are going to look at glycolysis, and it is defined as a biological process where glucose is converted into pyruvate to provide cells with energy. So we are breaking down glucose because now glucose is in excess. So the body has to look for ways on how to store our glucose. And at that point is when the glycogen synthesis also takes place, because the excess glucose can also be converted to glycogen for storage. So this process occurs in the cytosols of the cell, and that's why it's always very important to understand much about the cell and its organelles, the functions of those cell organelles, so we'll know where the cytosol is. So glycolysis takes place in the cytosol. And then another point here is that the intermediates which you are going to meet in the glycolytic pathway, there are only 3 carbon and 6 carbon atoms. So if you talk about 3 carbon atoms, if you go back to one of my sessions when I was introducing naming of monosaccharides, you will see what a 3 carbon is, and based on the functional group and the number of carbon atoms, you'll name it as either ketotriose or an aldotriose, and based on the 6 carbon atoms and the number of functional group, you'll name it as ketohexose or aldohexose. So the intermediates of the glycolytic pathway are either 3 carbon or 6 carbon atoms. Then there are 5 main categories of the reactions which take place in the glycolytic pathway. We have phosphoryl transfers. Phosphoryl transfers, always the class of transferases of enzymes will always do that work. So if you talk about the class of transferases, remember when we are defining enzyme classification in class, we said that they are involved in transfer of groups, specifically the phosphates. So we talk about the phosphoryl transfer, where the kinases are involved. That is one of the categories of the reactions which take place in glycolysis. We also have phosphoryl shift. Phosphoryl shift, shifting of a phosphate group, always the mutases will always work on that. So the mutases will always catalyze the shifting of phosphate groups. Then we have the isomerization reaction. The isomerization reaction, always the isomerases, and especially the functional isomers. We are going to see where a ketose is converted to an aldose to a ketose. So in that case, we talk about an isomerization reaction which will be catalyzed by isomerases. Then we have a dehydration, removal of water, and then lastly we'll have a reaction which is also involving aldol cleavage. Cleavage is where we are going to cut our 6 carbon atoms into 2 of the 3 carbon atoms, or an exose is broken down to 2 of the trioses. So these are the 5 main categories of the reactions we are going to see in the glycolytic pathway. In each of the 10 steps, we are going to see which of those reactions take place in each of those steps. In glycolysis, we have 3 stages. We have stage 1. In stage 1, we have to destabilize our glucose, D-glucose to be specific. Remember, we defined optical specificity of enzymes, and we said that in carbohydrate metabolism, optical specificity, the enzymes will only be specific on the D sugars. So in this case, we are talking about D-glucose. We are starting with D-glucose. Still refer to my sessions of monosaccharides. I explained well how we can draw D and L glucose and how we can draw alpha D glucose and beta D glucose. So we are starting with the D glucose. It means it is a linear form. Our D glucose is in a linear form. So in stage 1, we start with our D glucose, then we destabilize that D glucose. So that is the main objective of stage 1. We have to start with D glucose and destabilize it. Then stage 2, we have to generate the 2 trioses. So by splitting the hexose, the 6 carbon sugar, we are going to generate the 2 trioses. Then lastly, which accomplishes the main objective of glycolytic pathway, is we are going to generate energy in form of ATP. So glycolysis has the 3 main stages. Stage 1, we have to destabilize our glucose, D glucose to be specific. Stage 2, we have to cut the 6 carbon sugar into 2 of the 3 carbon sugars, or splitting the 6 carbon sugars into 2 of the 3 carbon sugars, and lastly we are generating energy in form of ATP. This slide shows all the 10 steps in glycolysis. So to start with, if you look at this is D-glucose. So step number 1, we start with our D-glucose. Remember, at the point when glycolysis takes place, there is excess of glucose, which has to be stored in form of glycogen, which has to be broken down to pyruvate with the main objective of providing energy in form of ATP. So our D-glucose, the first step, is an irreversible step. The first step is an irreversible step. Our glucose is converted to glucose 6-phosphate. The enzyme is hexokinase. When we are doing substrate specificity in class in relation to specificity of enzymes, we saw that the glucokinase was specific for glucose. But in this case, hexokinase has high affinity for glucose than the glucokinase. So the hexokinase, being a transferase, will transfer a phosphate to the 6th carbon atom of glucose, and only the 6th carbon atom will change by adding a phosphate group. So here we have a phosphoryl transfer. The hexokinase will do the phosphoryl transfer, and see, it comes in as adenosine triphosphate, 3 phosphates surrounding the adenine. Then it goes out 1 phosphate less because we are having an adenosine diphosphate. It means one of the phosphate has been bound to the 6th carbon atom of glucose to form glucose 6-phosphate. So this first step is irreversible. This first step is ATP utilization step. Look at how it goes in and where the arrow goes out to. So it comes in as adenosine triphosphate, meaning it is consumed. It is ATP utilization step. So it goes in as ATP, comes out as ADP. At that point, we talk about hexokinase converting D-glucose to glucose 6-phosphate. The kinases at their active site work well by the presence of magnesium ion or manganese ion. After conversion of glucose 6-phosphate, we are going to convert glucose 6-phosphate to fructose 6-phosphate. We are talking about stage 1. Step 1 to step 3 involves stage 1 of glycolysis. Step 1 to step 3 is stage 1 of glycolysis. We are going to destabilize our D-glucose to at last form fructose 1,6-bisphosphate. So step 1, 2, 3 are in stage 1. So from glucose 6-phosphate to fructose 6-phosphate, the enzyme which converts fructose 6-phosphate from glucose 6-phosphate is phosphoglucose isomerase. Most of these enzymes will always get the name from the substrate. It's not obvious, but most, I've said most of them. So in this case, the substrate is glucose 6-phosphate. It is a glucose already, which is phosphorylated, so the name phosphoglucose. Isomerization reactions. These are isomerases based on the International Union of Biochemistry Union. The International Union of Biochemistry gave 6 classes of enzymes based on the reaction they catalyze. So in this case, isomerases, it could be optical, functional isomers or geometrical isomers. But in this case, we are talking about functional isomers. So we are converting a ketose to an aldose to a ketose. So in that case, we are talking about isomerization. The enzyme is isomerase. The substrate is glucose 6-phosphate, which is converted to fructose 6-phosphate. So the name of the enzyme based on the reaction and the substrate is phosphoglucose isomerase. Fructose 6-phosphate is converted to fructose 1,6-bisphosphate. If you look at that step, it is another phosphoryl transfer. A phosphate is transferred to the 1st carbon atom to form fructose 1,6-diphosphate. 2 phosphate groups now at carbon atom number 1 and carbon atom number 6. So whatever is easier in these metabolic pathways is only 1 carbon atom is interfered with. When we started off with glucose, you know how to draw the D-glucose. Then all the carbons from number 1 to number 5 is the same. Only carbon atom number 6 changed by adding a phosphate. In the second step, you already know the structure of fructose, which you learned in Biochemistry 1. So in that case, in Introduction to Biochemistry, we were taught on how to draw D-fructose. So even after knowing the structure of D-fructose, you just add a phosphate at carbon atom number 6. Then we have fructose 6-phosphate. But now the third step, still the substrate is phosphofructose because it is fructose which is phosphorylated, but now the kinases are the ones which are doing the phosphoryl transfer. It transfers a phosphate group with the first carbon atom of fructose, and we will have fructose 1,6-bisphosphate. The 1 and the 6 shows the positions of the carbons where the phosphorylation has taken place. So at this point is also an ATP utilization step. The third step is also a reversible step. So still the kinase at step 3 will need either magnesium ion or manganese ion for it to be active or to catalyze that step. So in that case, we talk about an ATP utilization step. We are done with stage 1, destabilizing our glucose. At last, we are forming fructose 1,6-bisphosphate or diphosphate. So in stage 2, where we have to split the 6 carbon atoms into 2 trioses, it involves step 4 and number 5. So step 4 and 5 involve stage 2, where we are going to generate the 2 trioses. So we have the substrate being fructose 1,6-bisphosphate. Some books will write bisphosphate, some books will write diphosphate. The enzyme will be taking the name of the substrate, fructose bisphosphate aldolase, or just an aldolase. Even if you don't include fructose bisphosphate aldolase, even aldolase is okay. That is an aldol cleavage. Cleavage, we are cutting the ketohexose, which is D-fructose or fructose 1,6-bisphosphate, into 2 equal halves. Then we have a ketotriose and an aldotriose. So that is the aldol cleavage. It's going to form the dihydroxyacetone there, which has a keto group. It's a ketotriose, and down here it's going to form glyceraldehyde 3-phosphate, which is an aldotriose. From step number 6 to step number 10 is stage 3. The main objective of stage 3, we have to generate energy in form of ATP. So they talk about ×2, because now we have the triose. The dihydroxyacetone will go to lipid metabolism or to formation of lipids. We proceed with glyceraldehyde 3-phosphate, such that at the end of stage 3 we have 2 of the pyruvates produced. So it talks about ×2. So from glyceraldehyde 3-phosphate, it is converted to 1,3-bisphosphoglycerate. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. The only enzyme which is an oxidoreductase in glycolytic pathway is glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate, we have to add 1 phosphate or a PI. The enzyme is NAD+ dependent. What goes in is what the enzyme depends much on. It generates NADH. Very important. NADH is needed in electron transport chain. So it generates NADH, which will act as reducing equivalents. We are forming 1,3-bisphosphoglycerate. I still said that not much changes in this structure. So we have the second carbon, the third carbon remaining the same. Only the first carbon we have a phosphate and a keto group. So you talk about 1,3-bisphosphoglycerate. So we have 1 and third carbon having a phosphate group. So you're talking about 1,3-bisphosphoglycerate. That is step number 6. Then step number 7, where the first substrate level phosphorylation takes place. It means it is an ATP generation step. The first ATP generation step is by phosphoglycerate kinase. ATP is generated. We are forming 3-phosphoglycerate from 1,3-bisphosphoglycerate. So in that case, we have the first substrate level phosphorylation. ATP is generated, and I have to stress that that step is a reversible step. The reversible steps which we've met are only in stage 1. Step 1 and it's step 3. But now step 7 is reversible. The enzyme gets the name from the substrate, phosphoglycerate. It is a kinase. ATP generation step. 3-phosphoglycerate forms. From 3-phosphoglycerate, we are going to form 2-phosphoglycerate. So at that point is when we talk about phosphoryl shift. Phosphoryl shift. We are shifting phosphate from the 3rd carbon atom to the 2nd carbon atom. And shifting, mostly in molecular biology, talk about frame shift. It's always a mutation. So the mutase, an enzyme called mutase, will catalyze that step of formation of 2-phosphoglycerate from 3-phosphoglycerate. Then the 9th step is where dehydration process was taking place. We are forming phosphoryl pyruvate from 2-phosphoglycerate by removal of 1 molecule of water, the enzyme being enolase. We are going to form phosphoryl pyruvate. So in that case, enolase, since water is removed, it falls under the class of enzymes lyases. You know, lyases will always catalyze a reaction which ends up in formation of double bonds. So it is a lyase as a class of enzyme, but in this case we are talking about enolase specifically, which catalyzes the step, and 2-phosphoglycerate is converted to phosphoryl pyruvate. And lastly, step number 10, which is the last step in glycolysis in the formation of pyruvate, is irreversible. Pyruvate kinase catalyzing the enzyme, catalyzing the reaction, and then it is an ATP generation step. So kinases still need magnesium ion and manganese ion to be active or to catalyze a reaction. So in that case, we are forming 2 pyruvates for 1 glucose, which is broken down. So the process of glycolytic pathway of the glycolysis breakdown of glucose to pyruvate with the main objective of generating energy in form of ATP, it has 10 steps. And in these 10 steps, 3 of those steps are irreversible. Step 1, step 2, step 3, and step 10. Step 1, step 3, and step 10. Isomerization takes place at step 2 and step 5. Phosphoryl transfer takes place at step 1 and step 3. Substrate level phosphorylation takes place at step 7 and step 10. So in this case, we are talking about glucose, which has been broken down to pyruvate with the main objective of generating energy in form of ATP. So with the biochemistry pathway can be broken down to provide energy. At the time we are talking about glycolytic pathway and breakdown of glucose to pyruvate, we ask ourselves, what is the fate of pyruvate? So if we talk about the fate of pyruvate, we talk about aerobic and anaerobic. Aerobic is presence of oxygen. Anaerobic, then there is no oxygen. So in aerobic, it forms acetyl-CoA. In aerobic, it forms acetyl-CoA, not pyruvate as I written here. I'll just be able to change it. But in anaerobic, it forms lactate and ethanol. So if you look at in anaerobic, it takes place much in the microorganisms and the yeast where there is alcohol fermentation. And first, pyruvate is converted to acetaldehyde by decarboxylation process. And then, after that, it is further converted to ethanol. So the decarboxylase will reduce the pyruvate, the 3-carbon, to acetaldehyde, which is 2-carbon. Then acetaldehyde will be reduced to, will form ethanol by alcohol dehydrogenase. So in that case, at the point it's forming ethanol, the enzyme is NADH dependent and will be able to generate NAD+. Still, in anaerobic state, we'll be able to form lactate. It is a one-step. Pyruvate is converted to lactate. The enzyme is lactate dehydrogenase, and lactate is used as a fuel for cardiac muscle as well as the brain neurons. Then lastly, acetyl-CoA. Pyruvate is converted to acetyl-CoA, which is used in the TCA cycle or can also be used in lipid metabolism or, for example, in cholesterol synthesis. So a bit of medical importance of glycolysis. It provides energy to the cell. We've seen ATP generation steps, so it's a very important pathway. Secondly, the heart is mainly aerobic organ, so when there is myocardial ischemia, there is a decreased ability for those cardiac muscle, and ATP production is affected, and in that case the heart is affected, and you've seen the ATP generation steps. Still, erythrocytes rely much on the ATP produced, so the enzyme pyruvate kinase, if it's defective, then it leads to hemolytic anemia, which is also as a result of erythrocytes depending much on energy from glycolysis. So if there is deficiency of the enzyme pyruvate kinase, then there will be a problem with erythrocytes. Deficiency of phosphofructokinase, which was seen as an ATP utilization step for step number 3, if the deficiency of that enzyme takes place, then there is decreased muscular performance, which might lead to fatigue. Then another point is clear here, that fructose and galactose are also metabolized using glycolytic pathway. So after taking a component which has sucrose in it, digestion will take place, absorption will take place for glucose, but then for fructose, the same metabolic pathway, glycolytic pathway, that is how it is going to provide energy. Same with galactose. After taking a component of lactose, which is milk sugar, digestion takes place, absorption takes place, then galactose, it will enter glycolytic pathway. That's how it will provide energy. So it needs to have an intermediate of the glycolytic pathway for it to provide energy. So amphibolic role of glycolysis is very important because it provides intermediates or it provides building blocks for other pathways. For example, the dihydroxyacetone phosphate, which was produced at the step where we had the aldol cleavage at step number 4, very important because it is used much in the synthesis of triglycerols or in lipogenesis. The 3-phosphoglycerate, which was forming in step 6 from 3-glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate to 3-phosphoglycerate. So that product, 3-phosphoglycerate, which is forming from 1,3-bisphosphoglycerate, is used in the synthesis of amino acid serine. If we refer to serine, it was alcohol-containing amino acid. So 3-phosphoglycerate, one of the intermediates of the glycolytic pathway, is used for its synthesis. Pyruvate can also be used in the synthesis of amino acid alanine. If you talk about gluconeogenesis, which we are going to discuss, that is how alanine one-step reaction will be converted to pyruvate, or pyruvate can be converted to alanine. And that is how the amino acids act as non-carbohydrate precursors to generate glucose at time of starvation. Aerobic glycolysis also will provide us with acetyl-CoA, which will be used by